Preparation Model Question Paper 14. I'm Anand Shankar Reddy, PhD Plant Pathology. Demicyclic rust. So demicyclic means lack of uridial stage is generally considered demicyclic, and this question was very important by considering the previous question papers. If you see the previous net question paper, this question was very quite common asked question paper from the past 6-7 years. So demicyclic means lack of uridial stage is known as demicyclic. The second one is the TMV genome can be altered with tobacco mosaic virus. The genome can be altered with ADP, adenosine diphosphate. If you see in tobacco mosaic virus, that was a very important plant virus when it comes to plant virology. Because if you see majority of the first discoveries or majority of the breakthroughs, it is a completely model plant virus for plant virology. So generally the genome can be altered by using ADP, adenosine diphosphate. I think we guys know very well ADP. What is ATP? Adenosine triphosphate, adenosine diphosphate, which contains 2 molecules, whereas adenosine triphosphate which contains 3 phosphate molecules. So ATP is generally the form where we will get the energy. ADP in the sense if one phosphate molecule was lost, when it comes that goes to the state of ADP. Again, if the energy generated ADP becomes ATP, that is secondary. So here the TMV genome can be altered with ADP, adenosine diphosphate. The third one is a bread mold fungus. Bread mold fungus means what is the fungus that can generally grows on bread? That is a Rhizophus stolonifer. Generally all Rhizophus species like Rhizophus stolonifer, Rhizophus, various other species of Rhizophus which generally grows on bread. But the most commonly the fungi that we can observe on bread is Rhizophus stolonifer. The smallest plant pathogen in the world: virus, phytoplasma, viroid, and spiroplasma. So when it comes to all these groups of plant pathogen, viroid is the smallest plant pathogen in the world that we know so far. Virus is small, but viroid is 100 times smaller than that of virus. When it comes to phytoplasma and spiroplasma, that are more or less similar to the size of virus. Sometimes it is actually bigger the size of virus. But when it comes to viroid is the smallest plant pathogen that was reported so far. So it is actually 100 times smaller than that of virus. Imagine this is the virus. So the viroid is this, like this. So the viroid is the smallest plant pathogen that have been reported so far. So PCR can amplify. So generally PCR can amplify both the DNA and RNA. If I see the literature and the many books, it is mentioned that both the DNA and RNA. In the many books also they have mentioned the same, but as far of my personal experience, I have isolated DNA in my MSc, but the PCR can only amplify as far of my expectation. I don't know whether it can it can amplify RNA or not, but as far of my knowledge, it can amplify DNA. That's I know sure. But RNA I am little bit doubt about, but many of the books and literature that I have mentioned, it can amplify both DNA and RNA. If you guys know, please let me come to know. I'll be happy. Generally PCR can amplify both DNA and RNA. Etiology means, generally etiology means study of symptoms, right? So study of symptoms is generally known as the etiology. And the next one is the virus that infect the bacteria is known as. So bacteriophage, bacteriocin, bactericide, and any molecules. Generally a virus that infect bacteria is known as bacteriophage. So bacteria means we know very well general bacteria. Bacteriophage, phage means eaters. Bacteriophages are the bacterial eaters. If I want to tell you the definition, a virus that infect the bacteria is known as bacteriophages. And there was one more terminology, Dillo-Vibrio, right? A gram-negative bacteria that infect another gram-negative bacteria is generally known as Dillo-Vibrio. Generally, in bacteria that infect another bacteria is Dillo-Vibrio. But a virus that infect bacteria is known as bacteriophage. Bacteriocin means that is a proteinaceous substances, or I would say chemicals that is produced by the bacteria which kills closely related strains of bacteria. That is called bacteriocins, or you can say toxins that is produced by the bacteria that can kill closely related species. Bactericide means it's kind of, however we are using the general terminologies, fungicide, viricide, like that, a chemical which is used to kill the bactericide, but the commercial availability of bactericides are very less. And any molecules, generally bacteria is termed as any molecules, because when the Antonio van Leeuwenhoek first discovered this bacteria, he called it as any molecule. Later Ehrenberg in 1838 coined the term bacteria. Generally, Eli molecules means bacteria only, nothing else. The next one, methyl heptyl dinitrophenyl chronate, crotonate, that is generally dinocap. So the trade name of dinocap they're asking carthen. So carthen group of fungicide generally recommended for powdery mildews. You can see majority of the powdery mildews. This carthen group of fungicides are especially recommended. And the trade name of dinocap is again carthen. So Bravo, I think chlorothalonil, Bravo or Kavach, Botra and Dexon, some chemical names like trade names have been given here. And suicidal germinations are takes place in generally suicidal. If you see this Striga, Cuscuta, Warabanki, Loranthus, these are all the phanerogamous plant parasites. So generally suicidal germinations are found in Striga. So the next one is I think you guys know about Cuscuta, Loranthus, Warabanki and Striga. Cuscuta is a complete stem parasite. Loranthus is a partial stem parasite. Warabanki is a complete root parasite. Striga is a partial root parasite. This partial root parasite Striga is in where we can observe the suicidal germinations. Other names also been there for this phanerogamous plant parasites. So potato spindle tuber viride is generally transmitted by. So first of all, we need to understand potato spindle tuber is a disease that is caused by viride. You remember, as so far there have been 40 virides have been discovered. Might be excess also, as far of my knowledge. So far there have been 40 virides have been discovered. All virides contains genome as RNA. If I want to tell you single stranded RNA, all virides contains genome as single stranded RNA, and all the virides are transmitted by mechanical transmission only. There is no other means of transmission have been discovered in virides. The major and the most important transmission method is mechanical transmission or sap transmission, I would say. But generally, if you see viruses viruses can be transmitted by various kinds of agents, especially insects. And here the potato spindle tuber is a viride. Generally, potato spindle tuber viride is transmitted by mechanical transmission. As I said, not only potato spindle tuber, but all the plant viride diseases. Viride, we cannot, there is no necessity to include plant viride because virides are only meant for plants. So the next one is the select the correct pair from the following: hypertrophy, which means decrease in size. Sorry, hypertrophy. Hypertrophy, increase in size. Hyperplasia, uh, increase in number. Actually, the all the above are actually the correct pair. It is supposed to be hypertrophy, right? Yes, of course, it's right only. Hypotrophy, it's not hypertrophy. Hypotrophy, which means decrease in cell. Hypertrophy, increase in size. Hyperplasia means increase in number of cells. Here the correct pair is all the above, but it is supposed to be hypotrophy. And the next one is application of potash generally increases resistant to waterlogging, drought resistance, and as well as disease resistance. Generally, this potash is actually meant for 2D, disease resistance and drought resistance. So application of potash is actually beneficial for increasing the disease resistance and as well as the drought resistance. So the answer is B and C. Yellow mosaic of Bendy is spread by. So Bendy yellow mosaic virus is actually spread or transmitted by whitefly, Bemisia tabaci. VAM, which means vesicular arbuscular mycorrhiza, which is actually a fungi, bacteria, or virus. So it is actually a fungi. Then next one, teleospores of rust fungi have how many number of germ pores? If we see rust, we can in rust 5 spore stages are there, right? 0 stage, stage 1, stage 2, stage 3, and stage 4. So here the 5 stages, pycnium, asium, iridium, telium, and vesinium it will come. So in the next slide, I have placed like how many germ pores are produced by each spore stage. So here when it comes to teleospore is actually bicelled. In the only bicelled spore in the life cycle of a wheat stem rust is teleospore. So I repeat, the only spore stage or the only spore in the entire rust life cycle which is a bicelled, that is teleospore, and it contains the germ pore 2. So teleospores contains 2. And the next one is the brown leaf spot of rice is caused by Helminthosporium varese or Bipolaris varese. Here they have given Drusillera varese, Cercospora reticulata, Xanthomonas varese, Pericularia varese. Here, as I told you, brown leaf spot of virus is also called responsible for a Great Bengal famine. It is caused by Helminthosporium varese, otherwise called as bipolaris varese. The teleomorphic stage or sexual stage is Cuculiobolus meabanus, and it is can also called as Drusellera varese. So the answer is Drusellera varese. And Cercospora arachidicola causes Thicca diseases in groundnut. Xanthomonas varese, it causes many diseases in fruits as well as in rice also, bacterial blight of rice, which is caused by Xanthomonas varese. Pathor varese, right? And Pyracularia varese, we know blast disease of rice, which is caused by Pyracularia varese. And the next one is kernel bunt of wheat is caused by. So generally, the kernel bunt of wheat was first reported by Mitra in 1931. Why it is called kernel bunt? Because the disease was first reported in the kernel. So in kernel, there is an agriculture research station where they used to grow wheat. So in that, a new disease will occur, that is a bunt. So that was first discovered by Mitra. Since it is the area that kernel, so they named a kernel bunt because it was the first appeared in that kernel. So that was first discovered by Mitra in 1931. The Mitra named the disease as Tilicia indica. So later B.B. Munkur name changed to Neovesia indica. Again, couple of other scientists are started quoting why we need to rename it to Neovesia indica. Let's call it as a Tilicia indica as per of Mitra, because this why this Munkur reassigned this genera to Tilicia from Neovesia. When the Sporidia is producing, the characters of the Sporidia are much more similar to Neovesia. That's why B.B. Munkur placed this bunt fungus, that is kernel bunt, renamed it as a Neovesia. Later, various scientists and societies, based on only Sporidia, we cannot come to a conclusion. Let's as it is like a Tilicia indica. If we see the literature, both names will occur, can be found like Tilicia indica or Neovesia indica. The answer is Neovesia indica. Which fungicides give effective control of covered smut of barley? Vitavax, Serasan, Agrosan, and Sulfurdus. Generally, all smut disease can be effectively managed by Vitavax. Seed treatment with Vitavax. So Vitavax, so oxycarboxin, oxycarboxin, the trade name is Plantvax. Carboxin, the trade name is Vitavax. So this carboxin or Vitavax can be generally recommended for the seed treatment or the management of all kinds of smut diseases, not only this barley smut. And the next one is Plasmodium brassicae, that causes clubroot disease of crucifers, can be controlled by. So clubroot of cabbage, that was caused by Plasmodium brassicae. The life cycle was discovered by M.S. Voron in 1876, around 1878, I think so. So this M.S. Voronin, who discovered this clubroot of cabbage life cycle, is also a student of Anton de Berry. So he is not only discovered the clubroot of cabbage life cycle, but also discovered the life cycle of potato wart, Syncytium endobioticum. And when it comes to the management, so just this disease were very severe in low soil pH. If you say 5.2 or 5.3, the soil pH, the disease was very severe. If we just increase the soil pH by applying a lime, so we can efficiently control this disease. So the answer is just raising the pH of soil. The options is raising the pH, decreasing the pH, both A and B, none of the above. The answer is raising the pH. So if we raise the pH from 5.2 around 7.6 or 7.8, we can efficiently control the disease. So for raising the soil pH, we use a chemical called lime. Around 2.5 tons of lime can be recommended for 1 hectare to raise the soil pH. So just raising the soil pH is more than enough to control this disease very efficiently. The next one we have discussed this spore stages, right? Like teleospore, how many germ pore it will produce. If we see the spore stages of rust fungi, there are 5 stages are there: pycnospore, achiospore, iridospore, teleospore, and basidospore. Pycnospore generally assigned to stage 0 because actually this achiospore, iridospore, teleospore, these 4 stages were first discovered. That's why they named as 1, 2, 3, 4. But the pycnospore we cannot name it as a 5 because this is the most important sexual stage or the sexual spore where the other spores are coming out from. So if we see, that's why they named it as spore 0. So pycnospore spore 0, it will produce a germ pore 1. So achiospore stage 1, it will produce 6. Iridospore stage 2, it will produce 4 germ pores. Teleospore stage 3, it will produce 2 germ pores. That's what we have discussed in the question number 15. So basidospore at the stage number 4, it will produce only one germ pore. Here generally pycnospore look like hyaline, whereas achiospore yellow, golden brown iridospore, teleospore generally look like black, dark or brown color, black or dark brown color. Again basidospores are hyaline in color. When we see the nuclear conditions, pycnospores are haploid, remaining all pycnospores and basidospores are haploid. Remaining all spores are dikaryotic. Only teleospore is diploid. Since I told you, the only bicelled spore in the entire life cycle of wheat stem rest is teleospore only. So generally pycnospores and achiospores, these two stages happens in the leaf of barberry. Barberry is alternate host, right? So in the pycnospore generally happen in upper surface, whereas exactly on the corresponding lower surface, this achiospore will happen. If you see the iridospore that happen on the upper surface, and the teleospore is also side by side to the upper surface. When the crop matures, less number of iridospores are produced and more number of teleospores are produced. These iridospores and teleospores are produced on the primary host, that is wheat. The remaining two stages, pycnospores and achiospores, are that happens in the barberry. Okay, so this generally this basidospores actually in survives in soil debris in an active form. Sometimes it active when it requires. And iridium and teleal stage which occurs in wheat, that is primary host, whereas pycnial and spermatial stage, the spermatial stage uh, uh, the pycnial stage is spermatial. Pycnia is otherwise called as spermatia, so otherwise called as zero stage, otherwise called as sexual stage also. I repeat, pycnial stage is otherwise called as spermatial stage or zero stage or sexual stage, whereas the second one is acial stage. This both will happen in a barberry, that is an alternate host. White blister of crucifers is caused by the pathogen. So white blister of crucifers generally caused by albugo candida. So Pythian debarium generally causes damping of diseases, and Plasmopora viticola grapes downy mildew. This albugo candida which causes a blister blight of crucifers. Sclerospora sorgi which generally causes downy mildew in sorghum. Then sandal spike disease of sandal was caused by, and generally this sandal spike was a phytoplasmal disease, and it is the first reported phytoplasmal disease in India. It was first reported by Verma et al. In 1968, I think around 68 or 69. So this is the first reported phytoplasmal disease in India. So sandal spike is generally a phytoplasmal disease. Then again one J.C. Luthra and it associated developed a solar heat treatment for wheat seeds for the control of wheat loose smut. So if you see this solar heat treatment, the name itself indicates solar. By trapping the solar energy, by having the solar energy or light energy from the sun, we can kill this pathogen with loose smut which are present in the seeds. So this treatment is only effective against effective against especially during summer seasons where the summer temperature is high, and we cannot perform this in winter and other seasons. Okay, so it can only perform during summer seasons where the summer temperature is more than 42, in between 42 to 44. Above 42 is very much good for the solar treatment. So the solar heat treatment is especially recommended for loose smut of wheat. And a bacterium which have a large number of flagella all over the body, over the cell, is organized in. So here flagella all over the body is known as peritrichus. Atrichus means single flagella. Amphitrichus means single flagella both sides. Lopotrichus, like this in bacteriology we have mentioned atrichus. Uh, Atrichus means there is no flagella. A means no. Monotrichus means one flagella at one side. Amphitrichus means one flagella at both sides. So such kind of things I think we have studied in bacteriology. If you see examples, Atrichus xylella, Monotrichus xanthomonas, Amphitrichus, Peritrichus. The best example of Peritrichus is Irvinia. So the flagella all over the body, the best example is Peritrichus. So the next one is fungi and plant diseases was written by fungi and plant diseases was actually written by B.B. Munkur. So is the one who established Indian Phytopathological Society. And if we see this E.J. Butler, he is considered as father of plant pathology in India. He wrote one more book, that is a fungi and diseases in plants. Here fungi and plant diseases, that was written by B.B. Munkur in 1948. But when it comes to E.J. Butler, he wrote one more book called Fungi. He wrote actually many books, but the famous book is Fungi and Diseases in Plants, which was published in 1918. He is considered as father of plant pathology and as well as mycology in India. So J.F. Daster is the second president of Indian Phytopathological Society after S.R. Bose, and he's very famous for, or very renowned for, the establishment of Phytophthora genera in castor, that is seedling blight of castor, which is caused by Phytophthora parasitic, I think. So that was actually discovered by this J.F. Daster, and he is also the second president of Indian Phytopathological Society. The next one is select the correct order of events. Incubation, invasion. If we see in pathogenesis, generally attachment after attachment, infection, incubation, invasion, and colonization. This is the general procedure. The first must be infection. We can see the option number D. Infection, incubation, invasion, and colonization. This is actually the cycle that happens. If we see here, infection, incubation, colonization, and all those things, we can clearly see here. This is actually the order. Okay. Plant virus moves from cell to cell through plasmodesmata, lenticels, cuticles, and stomata. Actually, the plant virus generally moves from cell to cell through plasmodesmata, but here this plasmodesmata is actually smaller than that of plant viral cells, but still the virus is managing to move through, because the virus can produce a protein called movement protein. For example, if you see this is a plasmodesmata. This is actually the size of virus. Imagine, what I'm trying to tell is plasmodesmata is actually smaller than that of plant virus, but still this is the plant virus. It is actually bigger than that of plasmodesmata, but still this virus is moving through plasmodesmata. Then you can ask me why it is need to move through only plasmodesmata, because one cell to another cell it is easily connected by plasmodesmata. Again, if it want to infect another cell, again it need to come out, and again it go and enters into the plant cells through pinocytosis process. Again, it's difficult, right? So this plasmodesmata are the connections that can easily connect to the other cells or adjacent cells. So through this plasmodesmata, the virus can easily move from one cell to another cell. But this virus is actually bigger than that of plasmodesmata, but still it is able to move because it produces a protein called movement protein. This movement protein helps to move from cell to cell through plasmodesmata. So next one is viruses. Viruses are generally glycoproteins or lipoproteins or sulfoproteins or nucleoproteins. Viruses are generally nucleoproteins because it is a combination of nucleic acid and protein. If you see this virus, this is the components. It is a nucleic acid and protein code. So that's why it is called as combination of nucleoprotein, because it contains nucleic acid and protein. Rather than that, the virus contains nothing, right? Like not like a eukaryotic cell, like it contains ribosomes or some other things. The virus contains only two components, nucleic acid and protein code. So this Bowden and Pyre in 1936 generally called virus as a nucleoprotein because it is a combination of nucleic acid and protein. The next one is the molecules are sensitive to tetracycline, insensitive to penicillin, sensitive to penicillin, insensitive to tetracycline. Generally, this is sensitive to tetracycline. Again, it is also the option is like sensitive to tetracycline. It is also insensitive to penicillin also, but the options was a little bit here, but the answer is both sensitive to tetracycline but insensitive to penicillin also. But generally all molecules, if you see molecules like petaplasma and spiroplasma, if you see, they are highly sensitive to tetracycline and resistant to penicillin. The next one genome content of viroid is the genome of viroid. So I think in the previous slides we have discovered all viroids. There are so far 40 viroids have been discovered. All viroids contains genome as single stranded RNA. I would say positive single stranded RNA. All viroids can be spread through only mechanical transmission, and all viroids contains genome as single stranded RNA. RNA is the genetic material for all viroids. You can take any viroid. All the viroids contains genetic material as RNA. The next one, NETU viruses are. NETU means NETU viruses. Generally, these viruses are actually transmitted through nematodes. Actually forgot this. What is NETU? NEPU means nuclear polyhedral, some kind of things was actually forgot. This native viruses are generally are tobacco rattle virus, P. early Browning virus, both A and B, and none of the above. So the answer is both A and B. This tobacco rattle virus and Pearly Browning virus, both viruses are native viruses only. The next one, nucleic acid content of plant viruses are ranged from, generally all plant viruses contains 5 to 40% of nucleic acid and 60 to 95% of protein code. But there is an exemption called TMV. TMV contains 5% of nucleic acid and 95% of protein code. I repeat, all plant viruses contains 5 to 40% of nucleic acid and 60 to 95% of protein code. But there is an exemption called TMV, tobacco mosaic virus, which contains 5% nucleic acid and 95% protein code. So here the nucleic acid is the infectious material of plant virus, where the protein coat only protect the nucleic acid. No, protein coat have no other function, but majorly the culprit is nucleic acid only, which is actually causes the infection in all plant viruses. The next one is the sterilization temperature for autoclave. So the autoclave, the standard sterilization temperature for autoclave is 121.6. But generally, if you see in exams, they have also give 121, 15 minutes, 15 to 20 minutes for 15 psi. But the standard or exact temperature, sterilization temperature 121.6. In exams, sometimes they may give 121, 15 to 20 minutes for 15 psi. So that is also right answer based on the options given in the exam. But this is a standard or general one is 121.6 degrees Celsius. Vegetative stage of fusarium. So first of all, we need to understand this fusarium is a pathogen where it can produce three types of spores: microconidia, macroconidia, and chlamydospore. This microconidia, which means it contains no septations or sometimes one septa. Macroconidia contains 3 to 5, sometimes 7 also. If you see chlamydospores, chlamydospores are the resting spores. If you see the options, chlamydospores, rhizomorph, microconidia, and microconidium. Macroconidium and microconidium. This is not vegetative stage. If you see rhizomorph, this fusarium cannot produce rhizomorph. Another option is automatically chlamydospore. These are all the vegetative resting spores. Okay. Next one, wheat stem rust fungus. The Puccinia graminis enters into the plant cells through. So option is stomata, lenticels, trichomes, and hydatodes. So generally this Puccinia, wheat stem rust fungus, enters into the plant cells through stomata. So but some reports have been also there that this pathogen can enter into the plant cells through stigma also. In fact, the stigma also the entry point for this wheat stem rust. But since the option there is no stigma, the answer is stomata. If you see lenticels, trichomes, and hydatodes, you can also see the pathogens which generally enters. If you see hydatodes, black rot of cabbage, and rice bacterial leaf blight also, like that some other things will be there. And then the word fungicide is actually derived from, fungicide is actually a Latin word. So fungi, bacteria, virus, fungicide, these are all are the terms which are actually originated from Latin. So the next one is the X bodies. X bodies are actually produced by, X bodies are the inclusion bodies. Okay, include, what is inclusion body? Generally, for simply I will tell, it is like some kind of identification features that are left over by the virus. Each and every virus can leave different kinds of inclusion bodies. Here the X bodies are produced by tobacco mosaic virus. Here there are in the right side I have mentioned a couple of other inclusion bodies that are produced by the plant viruses. If you say tobacco mosaic virus, that is produced, tobacco mosaic virus produces X bodies, whereas cauliflower mosaic virus produces amorphous inclusion bodies, Zika virus produces granular inclusion bodies, rhabdovirus produces bullet shaped inclusion bodies, Poti virus produces pinwheel inclusion bodies or cylindrical inclusion bodies. So these are all the various kinds of inclusion bodies. But the question is, X bodies are produced by tobacco mosaic virus. On the right side we can also see here. And the next one is a monogenic resistance, vertical resistance, polygenes, horizontal resistance, vertifolio effect. So here monogenic resistance is generally the terminology is meant for vertical resistance because it is governed by single gene. Single means monogenic. So it is related to vertical resistance. If it's come to horizontal resistance, that is governed by multiple number of genes. So the vertical resistance is monogenic, where the horizontal resistance is polygenic. Okay. When it comes to vertifolio effect, so the loss of horizontal resistance while breeding for vertical resistance. If you are breeding for vertical resistance, or automatically the horizontal resistance will be lost. So that is called vertifolio effect. The concept was first identified by van der Planck. Okay, the next one is the fragmentation of specialized hyphae. Very simple. Imagine this is a hyphae. This hyphae is divided into small fragments. Is a asexual vegetative kind of reproduction. So we have published a couple of mycology videos regarding this. So if you see, each single fragment will divide and starts to behave like a new hyphae or new fungi. That is called this is generally called as oidea or arthrospore. The answer is both A and B. So the fragmentation of specialized hyphae gives arthrospores or idea. Arthrospores otherwise called as idea. Nothing much here. And the next one is the book entitled with fungicides in plant disease control was written by Y.L. Neen and P.N. Tapiliel. In the previous questions also, I think this question was there. Generally, fungicides in plant disease control was written by Y.L. Neen and P.N. Tapiliel. This was a classic book. I think the fourth edition was running now. The book was written by Y.L. Neen and P.N. Tapiliel. The first edition was published in 1971, I think. But the answer is fungicides and plant disease control was written by Y.L. Neen and P.N. Tapiliel. Kitagen and Hinosan are the fungicides which are used for the management of paddy blast, early blight of potato, cereal rust, brown spot of rice. Generally, this Kitagen and Hinosan are especially recommended for paddy blast. So not only this Kitagen and Hinosan and other two fungicides, this is generally the trade names Kitagen and Hinosan. Uh, Edifenfos and one more fungicide was generally there are common names of this Kitagen and Hinosan. And this paddy blast, one more fungicide is also there, specific fungicide, that is tricyclazole, that can be recommended for the management of this paddy blast, and application of silica and various kinds of resistant varieties also there. But this Kitagen and Hinosan, Edifenfos and iprobenfos, yes, Edifenfos and iprobenfos, the trade names are Kitagen and Hinosan. That can be effective in this paddy blast. The next one is stripe rust of wheat is also known as brown rust, black rust, yellow rust, and stem rust. On the right side I have mentioned, so stem rust is otherwise called as black rust. Generally the name black rust is denoted to stem rust. So stem rust and black rust are same. Second one is the leaf rust. So leaf rust is otherwise known as orange rust or brown rust. So the brown rust is also denoted to leaf rust. So the last one is stripe rust. So stripe rust otherwise called as yellow rust or sheath rust. Here the answer is stripe rust of wheat is also known as yellow rust. The next one is each Cleistothecium of Podospora lycostrica contains. So each Cleistothecium of Podospora lycostrica contains one ascus. Generally, in Ascomycota group of fungi, each ascus contains 8 number of ascospores, but in Podospora lycotrica contains only one ascus. Ascus means it is a compact mass of structure, or it's a I would say some kind of a structure where the spores are packed inside. Generally, you can see like a balloon kind of structures. It would be like this, like this, or maybe sometimes like this also. Where the spores are packed inside. Each ascus contains 8 number of ascospores. Generally, each fungi contains thousands of asci, where thousands of ascospores are packed inside the asci. Actually, generally hundreds of asci will be there, and inside the asci, ascospores are present. But there is a fungi called Podospora lycotrica, which generally causes rose powder mildew, I think so. This pathogen produces only one ascus. The ascospores are actually packed inside the ascus. Okay, each ascus of Ascomycota group of generally contains 8 number of ascospores. Taphina deformans, which generally causes, so Taphina deformans, which is caused by peach leaf curl. So peach leaf curl is caused by Taphina deformans. There are a couple of pathogens: Taphina epiphylla, Taphina maculans, like turmeric leaf blotch, Taphina epiphylla, strawberry, some kind of diseases. Okay, generally this Taphina deformans is responsible for peach leaf curl. It produces sexual fruiting body is known as the naked assay. And the theorem is generally used for a seed treatment fungicide, that is a seed dresser. It is not I mean, very rare cases may recommended for foliar spray. It's not at all a soil applicant and growth promoter, and foliar spray is also very rare as far of my concern. And it is a seed treatment fungicide, best known for seed treatment fungicide. And this serum was the fungicide which was first time discovered by Tisdale and Williams in 1934. And it is a very famous seed treatment fungicide since it's almost 80-90 years since the discovery. Still we are using the theorem from that time. It was actually discovered by Tisdale and Williams in 1934. Which one is not related to other? So cleistothecium, perithecium, apothecium, and trichothecium. So here this cleistothecium, perithecium, apothecium are the sexual fruiting bodies, but trichothecium is not a sexual fruiting body. Tricho means hair generally. So generally cleistothecium is a completely closed ascocarp. Cleistothecium, apothecium completely opened ascocarp. Perithecium partially opened ascocarp within an opening called ostiole. Naked as there is no ascocarp. And this will be there, but which one is related, not related means this trichothecium only. The next one is seed potato is not transported from Darjeeling area to other parts of the country because of a domestic quarantine due to potato wart. So potato wart is an endemic disease in Darjeeling. So if you see what is epidemic, endemic, pandemic, and sporadic, the best example came endemic is the disease is confined to a particular area or locality. The best example is potato wart. So this potato wart and this Synchytrum endobioticum is generally more prevalent in Darjeeling district. So that's why the potato that is grown in Darjeeling district or Darjeeling of Shimla is not sorry Darjeeling of West Bengal is not supposed to transport from other parts of the nation, so just because of a domestic quarantine. And the next one is carborundum powder, chemically known as, generally carborundum powder is chemically known as silicon carbide. So this silicon carbide is uh, chemically called chemically known as carborundum powder is chemically known as this silicon carbide, generally used in local Asian assays. By performing local Asian assay, we use a chemical called this carborundum powder. That chemical name is silicon carbide. Pseudothecium producing fungi, Taphrina deformans, Venturia inequalis, Claviceps fusiformis, and Protomyces macrosporus. Generally, this Venturia pathogen can produce pseudothecium where that can survive on the leaf. So this pseudothecium can the general leaf fitness period is around 8 to 10 hours for this apple scab, exactly 9 hours if I want to tell exactly. This pseudothecium is actually produced by Venturia inequalis, and naked as a clavis Taphrina deformans, and this claviceps generally produce sclerotia, right? And trichogyne is produced by, trichogyne is an tube-like structure or that formed during reproduction, right? Gametangial contact, right? Gametangial contact or gametangial copulation. I think gametangial copulation from ascogonium to oogonium. From there is a small tube formed that is called trichogyne or terebrator generally. This can be actually produced by ascogonium. Ascogonium means I would say like a male reproductive organ. Antheridium is a Antheridium is a male reproductive organ. Ascogonium is a female reproductive organ. Why it is called ascogonium? Because if it is formed in Ascomycota group of fungi, it is called ascogonium. If this male reproductive organ comes under oomycota, it is called oogonium. Based on the classification, the name of female reproductive organ changes here. Generally, this trichogyne is produced by females, sometimes males also, and the cinema is produced by Pycnidium, Sporadium, Acerulae, cinema. There are 4 kind of asexual fruiting bodies that is produced by the fungi. Generally, this cinema is produced by Ceratocystis, Graphium, and Bryosia. All the above answer is generally, if you see here Pycnidium, it is generally hollow globe flask shaped fruiting body produced by Macrophomina, Botrytiplodia, Phoma, Phomopsis, Philosticta, Ascogata, Septori. They are all that can produce Pycnidium. When it comes to Sporadium, Fusarium, Epicoccum, Stromella, Tubercularia, Volutella, and Myrothecium, these are all produced Sporadium. When it comes to Acerulum, the best example is Colrotricum, Peslotia, and Gliosporium. Coming to cinema, cinema is otherwise called as Corimium, Ceratocystis, Graphium, Arthrosporium, Isaria, and Harpographium, Trichorus. These are all the examples that can be produced by cinema. So if you see here, Ceratocystis, Graphium, these are all actually produced. Cinema produces all kinds of these things. So the example is answer is all the above. Osteolate ascocarp is known as. So cleistothecium is a completely closed cleistothecium. Apothecium is completely opened. Perithecium means like it's a flask shaped structure where a small ostiole or small opening is known as ostiole. So the osteolus ascocarp is known as perithecium. And the next one is the dictyospore means, so spore with both vertical and horizontal septa is known as dictyospore, or we can also call as pleospore. The best example is alternaria. If you see alternaria conidia, it exactly sometimes it look like the bell shaped, right? Sometimes it look like a bell shaped with a very long beak. Actually the beak is sometimes 3 to 4 times bigger than that of spore, right? So this dictyospore, which generally means uh, the spore which contains both vertical and horizontal septa is known as dictyospore. Best example is alternaria. Next one is bunt spore produces. This bunt spore generally produces which shaped sporidia? S shaped, T shaped, V shaped, H shaped. Generally the bunt produces H shaped sporidia. So the majorly H-shaped sporidia will be produced, especially if you see Tilicia and Neovesia, they produce this H-shaped sporidias. And coprophilous fungi means the fungi which are actually growing on the dung is known as coprophilous. Copro means it is a growth factor generally. So this coprophilous fungi means a fungi which actually favors in habitat of dung. So the answer is a fungi growing on animal dung, fungi growing on animals, fungi growing on human tissues, fungi going on dead tissues. So the answer is fungi going on animal dung. Generally, if I want to say horse dung is the exact answer. So the next one is give an example of soil-borne downy mildew. So the best example for soil-borne downy mildew is Kambu downy mildew. Here I have given sorghum, maize, Kambu and ragi. The best example is Kambu downy mildew is an example of soil-borne downy mildew. Founder of plant virology. Yeah, Adolf Meyer is generally considered as no, just a minute. Bizarnik, yes, he is also considered as a father and founder of plant virology, because Adolf Meyer and as well as Ivanovsky, they made a very breakthrough contribution. There is no doubt about it. But the founder of plant virology is Bizarnik only, because he is the one who coined the term virus, and is the first one who discovered tobacco mosaic virus also. That's why he's considered as father and founder of plant virology. Next one, the trade name of the fungicide triadimifon. So triadimifon trade name is Bileton. Bileton is the trade name for triadimifon. Other have been given Beam. Beam is tricyclazole trade name is Beam or Bin. Baker, Bileton, Baker. But the trade name of triadomorph is Bileton. The next one is the resting spore produced by Fusarium species. I think in the couple of slides before we have discussed the Fusarium produce three kinds of spores: Microconidia, Macroconidia, and Chlamydospore. These are all the three kinds of spores that is produced by the Fusarium species. So here the option is the Conidia, Sclerotia, Stroma, and Chlamydospores. Of course, Chlamydospores. And the next one is the Conidia. Of course, it also produces Conidia, that is Microconidia and Macroconidia, but they have not mentioned micro and macro. So I'll go with the Chlamydospore. The answer is Chlamydospore. The next one is Phytoplasma are sensitive to Phytoplasma. All Phytoplasma and Spiroplasmal diseases are always sensitive to tetracycline. So they are highly sensitive. Sorry, resistance, right? So they are resistant to tetracycline. They're resistant to penicillin, sensitive to tetracycline. They are highly sensitive to tetracycline, but they are highly resistant to penicillin. So penicillin drugs are not effective against the phytoplasmal diseases. The next one, osteolate ascocarp is known as, I think this question was already came. Osteolate ascocarp is known as perithecium. The next one, DPPQS, Directorate of Plant Protection, Quarantine and Storage, is located at Faridabad, Haryana. So this DPPQS actually started in 1947, I think. It was located in Faridabad, based on Faridabad. So all the guidelines related to plant quarantine that was given by this DPPQS only. So Robigolia was the festival that was initiated by the Romans against smuts, blights, rust, both A and B. Here, generally, the Robigolia is the festival that was initiated by a Roman king called Numa Pompilius in 700 BC. So Numa Pompilius is the king who initiated this festival called Robigolia. They used to worship to protect their crops against rust diseases. So the answer is rust. So nowadays we used to celebrate, not we, Romans used to celebrate on April 25th of every year. So the next one is in a long cycle rust, zero stage refers to. In a long cycle, in the sense, in the life cycle of rust, not long cycle of rust, is supposed to be life cycle of rust. So zero stage, I think we have already seen the 5 spore stages: pycnium, acium, iridium, telium, basidium. So the answer is I think we have discussed, right? So pycnium is the 0 stage, acium 1, iridium 2, basidium 4, acium, iridium, telium, and basidium, something like that will come. So the 0 stage or sexual stage or the spermatial stage is 0 stage, that is pycnial stage. The next one is fungi and diseases in plants was written by. This was very important question. If you see fungi and diseases in plants or fungi and plant diseases, we can see somewhere. Fungi and diseases in plants was written by E.J. Butler. Fungi and plant disease was written by B.B. Munkul. Fungi and diseases in plants in 1918. Fungi and plant diseases 1948. Pasteurization temperature for milk. Generally, I might have mentioned this milk, right? Because the sterilization temperature, pasteurization temperature may varies with drinks, but generally it's 62.8. Which of them are poisonous mushroom? Agaricus, Valvariella, Pleurotus. So these are all are edible mushrooms, right? Agaricus bisporus, Valvariella, Valvesiana, Pleurotus, Sozarkazu. These are all edible mushrooms. So the Amanita only the mushroom where it is poisonous. So this common name of the mushrooms, like what is agaricus mushroom, what is this common names that was mentioned in the next slide. These are all some edible species of majorly available mushrooms that are cultivated all over the world. So the padistra mushroom, or otherwise called as Chinese mushroom, Valvarella valvaceae, and edulous or hot mushroom, Agaricus bitrocus, and white button mushroom, that is Agaricus bisporus, and milky mushroom or summer mushroom, Calocybe indica, shiitake mushroom, Lentinus edodus, oyster mushroom, generally Pleurotus sozarkazu or Pleurotus species, because various species of Pleurotus generally comes under this oyster mushroom. Then molia disease of wheat and barley is caused by. So this molia disease is actually a nematode which is caused by Heterodera avinae. The next one is mycoplasmal diseases on plant was first reported by. The first phytoplasmal disease was reported by Doe et al. In 1967, and the first phytoplasmal disease is mulberry dwarf. We know very well. So the first reported mycoplasma, who reported, what they asking is who reported the mycoplasma for the first time, that is phytoplasma. Doe et al. 1967. Answer is Doe et al. And wheat rust life cycle India was first given by. In India, about wheat rust was actually first studied by K.C. Mehta, which is generally considered father of Indian rust, right? So Nagarajan later discovered this Paxinia pathways and other kinds of things that was discovered by Nagarajan. But in India, the first person who studies about this life cycle of stem rust is K.C. Mehta. And the next one, most destructive phase of the bacterial blight of disease is known as crescent phase, otherwise called as wilting phase. It's the most important phase. If you see the bacterial leaf blight of rice, which is caused by Xanthomonas varaceae, Pathovar varaceae, there are 3 stages can be seen: blight stage, crescent stage or yellow stage, the last one is yellowing phase. So in these 3 stages, the most important and destructive stage is crescent phase or wilting phase. The next one is the fruiting bodies produced by the fungus Ascochyta rabi is known as. So I think in the previous also we have seen like different kinds of fruiting bodies and all those things. Generally this Ascochyta rabi produces pycnidia. And give an example of enveloped viruses. Generally, if you see all the virus, Gemini virus, it's a twin particle virus. Potivirus, it's the largest group of plant viral family, right? And luteovirus. Among that, tomato spotted wilt virus is actually enveloped virus. It lengths about 100 nanometer. Even though it's 100 nanometer, somehow nobody are calling it as the longest virus. Wound tumor virus is generally people calling longest virus, even though it's a 70 nanometer. But if you see this tomato spotted wilt virus, its length around 100 nanometer. But there might be a reason behind the people are not calling this is the longest virus. Probably because of this enveloped nature. But when it comes to the example, which of the following is an example of enveloped virus, that is a tomato spotted wilt virus only. And the next one is aflatoxin that is produced by the strains of Aspergillus flavus. So there are a couple of strains are there in Aspergillus: B1, B2, G1, G2, M1, M2, like that. So if you see among all the strains, B1 is the most toxic and most destructive strain. So because if the food contains this B1 strain, it ultimately leads to death of the people. The remaining, not all the people but also animals. Among the strains, this B1 is very toxic and very dangerous. Next one, clamp connection is most common in generally this clamp connection is most common in smuts. And protein content of all plant viruses, I think we have already discussed. Majority of the plant viruses contains 5 to 40% of nucleic acid and 60 to 90% of protein code. 5 to 40% is the general nucleic acid content, 60 to 95% the general protein content that contains almost all plant viruses. But there is an exemption called the tobacco mosaic virus, which contains 5% of nucleic acid and 95% of protein code. Here they are asking protein content of plant viruses. All plant viruses generally ranging from how much they are asking. So generally all plant viruses contains the protein content of 60 to 95. So the answer is B. So the next one is antibodies are secreted by generally. So the antibodies are actually secreted by B lymphocytes and erythrocytes. I think it's a B lymphocytes. I think so. Probably I'm not sure about this question. Please go through yourself. I think antibodies are secreted by B lymphocytes can generally produce antibodies, and sometimes erythrocytes also. I'm not sure. Okay, the next question is the dilution point of TMB 1:1. Generally, the dilution point of TMB is 10 to the power of -6, which means 1:1 like here. The next one is tick out the wrong pair. Endemic. So endemic means I think there are four terminologies in epidemiology: epidemic, endemic, pandemic, and sporadic. Epidemic is otherwise called as epiphytic. Okay. Tick out the wrong pair here. Epidemic means the disease occurs like epidemic is otherwise called epiphytic, a sudden outbreak of a disease, so which where that can be present in all over the world, sometimes in a particular location also. Endemic means that confined to a particular country or district or location, but it is present in only particular place. Sporadic means irregular intervals it will come, and pandemic means that is present all over the world. Here they have wrongly mentioned option is pandemic. This is not prevalent in the country or continent or world, but the pandemic is generally the disease is present in all over the world. It is called a pandemic. So the wrong answer is pandemic. The downy mildew disease of pale millet is primarily, I think this was already came right. So generally the downy mildew of pale millet is generally a soil-borne pathogen. Head smut of sorghum is a head smut of sorghum. I think this came right, soil-borne just now. The next one is the cell wall of mycelium of true fungi contains. Generally the fungi contains chitin in their cell wall. Bacteria contains uh, mucopeptides are peptidoglycan. Viruses contains no protein coat, no cell wall, but generally protein coat act as a protecting structure. Here the cell wall composition of true fungi. Generally the fungi contains chitin and a little amount of glucan also. And glucan plus cellulose, glucan plus peptidoglycan, and a couple of other things also there. In the right side I have one small thing will be there. The general cell wall compositions of different plant pathogenic organisms. Fungi contains chitin and a little amount of glucan in their cell wall. Zygomycota cell wall is chitosan chitin or chitin chitosan. Oomycota cell wall is made up of cellulose plus hydroxyproline, little amount of hydroxyproline, but both. Hypochitoidiomycota cellulose plus chitin. Yeast cell wall is mannan beta glucan. Algae cell wall is glucan plus cellulose. Trichomystis cell wall is polygalactosamine galactron. Bacterial cell wall peptidoglycan or mucopeptide. Virus cell wall contains no cell wall, but this protein coat acts as a protecting structure. So this is about the cell walls. So the answer is B, glucan plus chitin or chitin. The teleomorphic stage of Cercospora. So Mycospirilla is the teleomorphic stage of Cercospora. Glomerella is for Coldotricum, I think. And other Pleospora, Cochiliobolus, these are all Cochiliobolus bipolaris helminthosporium. And other teleomorphic stage is also given here, but generally the Cercospora teleomorphic stage is Mycospirilla. So parasexuality was first discovered by Ponticarvo Roper, Erickson, D. Barry, Robert Koch. So Robert Koch is not at all related to mycology. Forget about him. Erickson, physiological specialization of fungi. D. Barry, father of plant pathology. But only one who left is Ponticarvo and Roper. The answer is Ponticarvo and Roper. They were the first who discovered a new phenomena that is called parasexuality in fungi. Parasexuality means plasmogamy, karyogamy, and meiosis not occurs in a regular stage at a specified intervals at specified manner is called parasexuality. That was first discovered by Ponticarvo and Roper in a fungus called Aspergillus nidulans in 1952. It is very important. Next one is given example of virus containing double-stranded DNA. Calimovirus, Gemini virus, Tobra virus, Lutiovirus. So the best example of double-stranded DNA virus, Calimovirus or cauliflower mosaic virus. The next one is protein subunits. Protein subunits, actually capsomer. Actually, the question itself is wrong. It is supposed to be the amino acids, the number of amino acids which are present in the TMV. So generally TMV contains 158. On the right side I have mentioned the TMV generally contains 158 amino acids. 2130 capsomer subunits are code protein or protein subunits. 6400 nucleotides. So here the uh, uh, questions, the answer for the question protein subunits means it's 2130, but since that option was not there, it is supposed to be the amount of the number of new amino acids which are present, but is actually the answer is 158. But the question is it is supposed to be the amino acids. So if it is a protein quota or protein subunits, it is supposed to be 2130. The computerized disease assessment training program. So distrain is the first computerized assessment training, not first computerized. Which of the following is a computerized means the distrain. Lopat is a technique that is meant for pseudomonas. It is used to identify the pseudomonas. Das ELISA, this is a technique which is identify the virus, and PCR is also same. That is technique used for the identification of fungi, bacteria, and virus. Next one is the virus enters into the plant cells through the process of so if you see the cell, they will just enters into the process of pinocytosis, which means they just absorbs cell, generally enters into the cell. If you see the videos of this virus entry, generally absorption kind of mechanism, you can see that process is generally called as pinocytosis. Sugarcane ratoon stunting is caused by, generally sugarcane ratoon stunting is um, caused by candidatus Phytoplasma, Virid, Xanthomonas or Lifesonia xylem subspecies xylem. Actually, it is caused by Lifesonia xylem subspecies xylem. It is a xylem limited it is a fastidiously vascular bacteria. And if you see this Lifesonia xylem subspecies xylem, previously called as Clavibacter xylem subspecies xylem, generally it causes sugarcane ratoon stunting disease. And hyperplasia means options is increase in cell divisions, cell death, abnormal enlargement of cells, and nanism. Nanism means dwarfism. Generally, hyperplasia means increase in number of cells. Hypertrophy, abnormal enlargement of cells. Hyperplasia means increase in number of cells or cell divisions. The next one is given example of the answer is increase in cell divisions or number of cells. An internally seed-borne fungal diseases, smut of ragi, sugarcane, red rot, jawar smut, grain smut, and the loose smut of wheat. Loose smut of wheat is the best example for internally seed-borne pathogen. Very, 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 very important. Not only this wheat loose smut, barley loose smut is also an example of internally seed-borne pathogen. But majority of exams, if you see, this wheat loose smut only we can see. The next one is Moco wilt of banana is caused by Fusarium oxysporum, Rhadophilus similis, Ralstonia solanacearum, Pentalon nigronervosa. First of all, we need to understand Fusarium is caused by fungi. Rhadophilus is a nematode. Pentaloria nigronervosa is the scientific name for this vector, aphid vector that is actually transmit this banana bunchita virus. This actually Moco wilt is caused by banana, that is caused by a bacteria Ralstonia solanacearum. The answer is the Ralstonia solanacearum. This Fusarium oxysporum pharma species cubens. Actually, this letter is supposed to be small letter. Subspecies cubens which causes Fusarium wilt or Fusarium wilt of banana. Rhadophilus similis, banana burrowing nematode, Pentaloria nigronervosa, which is actually the black aphid. The scientific name of black aphid vector which actually transmit banana bunchita virus. Banana bunchita virus is transmitted by black aphid, Pentalonia nigronervosa. The next one, the number of races of Phytophthora which are prevalent in India, 82. I am studying the same question from past 10 years. I don't know whether this was raised or not, like a number was raised or not. But as far of now, it is 82. I don't know, maybe updated information also might be there, but it was there in a previous model questions. The next one is effective fungicide for the control of rice blast. So tricyclozole is the recommended fungicide or effective fungicide recommended for rice blast. Tricyclozole. The trade name is Beam or Bin or Blastinius. These are the various kinds of trade names. Not only this tricyclozole, edifenfos or iprobenfos also can be recommended. I think in the previous question we have seen. But as far as the options given here, the answer is tricyclozole. The next one is Cleistothecium containing several asci bearing the defined rigid shaped appendages with coiled tips is the characteristics of A. Ansinella, B. Erisife, C. Sparotheca and Levilula. So here the answer is coiled itself, it is called Ansinella. To understand or to identify this, first of all we need to divide into two things. First was one ascus type and two ascus type. If you see here one ascus type here, if you see here one ascus type, myceliaid appendage, normal appendages only. One ascus with a normal myceliaid appendages means Sparotheca. If you see here some kind of uh, like branches like appendages, we can see here that one ascus with the branch like appendages is Podospera. Ansinella, if you see just now, we see coiled appendages. Cleistothecium with coiled appendages with many ascus Ansinella. The many ascus bulbous appendages. Bulbous at the starting we can see bulb-like structure that is called a bulbous appendage. The example is phylactinia. And many ascus with myceliaid appendages means single ascus means Sparotheca. Many ascus with myceliaid appendages means Erisife. We can see some kind of branch like appendages with many ascus. Single ascus means Porospora. Many ascus means this Microsporia. These are all the easy things for identification. The next one, Candida's cytoplasma contains, the cytoplasma generally contains genetic material as both DNA and RNA. DNA plus RNA is the answer. Then the next one is systemic fungicide used for the control of powdery mildew diseases. So all the powdery mildew diseases will be effectively managed by a fungicide called Calixin. So Calixin, Vitavax is generally that smut related smut related fungicide. MBC, that is mycozib, carbonism, I think, I'm not sure. The sulfur dust. This Calixin is especially recommended for this powdery mildew diseases. So bacteriophage is a bacteriophage means eater. Bacteriophage means bacterial eater. Naked single stranded RNA particle, virus infecting bacteria, virus infecting eukaryote cell, a animal virus. We know very well a virus that infects bacteria is known as bacteriophage. Reserve food material of yeast. So here there are a couple of reserve food materials. There is if you see here we can mention the I think in the next slide I have mentioned the list of reserve food materials of various kinds of organisms. So what is the reserve food material of fungi? What is the reserve food material? But when it comes to here, the answer is volutin granules is the reserve food material of yeast. The next one is the bacteria is the causal agent of a plant diseases was first reported by Cobb, DeBary, Mayer, Burrill. If you see, Cobb is a nematologist, Anton DeBary, we know, father of plant pathology. Adolf Mayer is a virologist. But when it comes to Burrill, T.J. Burrill is the first one who first reported the first plant bacterial disease, that is a fire blight of apple, which is caused by Yersinia amylovora. So previously it is called as Micrococcus amylovirus, later name changed to Yersinia amylovora. This is the first plant bacterial disease that was reported by T.J. Burrill in 1878. The next one is alternate host for Puccinia glumarum, Barbarus vulgaris, brinjal, Oxalis corniculata and Mahonia. So for Puccinia glumarum, the alternate host is Mahonia. Now, since as we discussed in the question number 98, the other storage organs of plant pathogens also mentioned here. Fungi generally store food storage organ is glycogen or oil in the form. Oomycota, mycolaminarin, algae, laminary, yeast, volutin. That is the question. So the yeast food storage organ is volutin. But when it comes to bacteria, it is also very important. In 2023 also this question was asked. What is the food storage organ of bacteria? Poly beta hydroxybutyrate. Sometimes they mentioned only beta hydroxybutyrate. That is also right. Okay. Poly beta hydroxybutyrate is the food storage organ of bacteria. So these are all the various kinds of food storage organs, and the answers have been given here for your reference. Might be one or two mistakes also might be there. You just correct it out yourself, and please let me know also if there is any mistakes. I will make it right next time. So what we discussed here is just a sample, and much more information about various kinds of aspects that was mentioned in my book, A Vision into Plant Pathology, a student version. For other kinds of guidance or any doubts, students can reach us at www.geekyresearch.com. Stay geeky and stay tuned. We are Team Geeky Researchers.